vamos a comenzar este acto que tiene por misión hacer académico correspondiente extranjero al profesor Silva, da Silva, y va a hacer la presentación doña Carmen Nájera, que es académica numeraria de la sección de Física y Química. Quería decir que en esta academia tenemos mucho afecto y mucho respeto a los académicos correspondientes, extranjeros y no extranjeros, pero los extranjeros, y para dar una idea del nivel, un poco presumir de la academia, el último, antes de usted, fue el profesor Stanley Whittingham. Quiere decir que eso indica que tenemos un nivel medio alto, y ahora hoy va a subir otro poco. Así que doña Carmen Nájera se servirá a presentar al recipiendario. Muchas gracias.
Muchas gracias, Miguel Ángel. Bueno, para nosotros es un honor que nuestros académicos correspondientes extranjeros nos visiten, porque no es tan sencillo como los correspondientes nacionales. Y entonces, claro, tienen que hacer el esfuerzo de venir, y ese esfuerzo puede ser mucho más grande si además, como es el caso del profesor Silva, tiene muchos cargos en su universidad. Y, de hecho, actualmente es el vicerrector de investigación, con lo cual aún le agradecemos más el que haya venido a Madrid a dar una charla sobre su investigación. En principio, él no ha dejado de investigar, o sea, él compagina toda su carga de gestión con la investigación, lo cual le ennoblece, ¿no? Y lo primero que voy a contar es un poco cómo ha sido su trayectoria. Él viene de la Universidad de Aveiro, es allí donde estudió Químicas, acabó en el año 87, leyó la tesis en el año 93, después pasó por todos los puestos intermedios de asistente, vamos a traducirlo como asistente, como titular y como catedrático. En el 2001 fue nombrado catedrático y ha desarrollado toda su carrera científica en dicha universidad. Ha pertenecido y pertenecen todavía, realmente fue presidente de la Sociedad Portuguesa de Química durante 2 mandatos, desde el año 2016 hasta el año 2023, y además es fellow, pertenece o es académico de la Academia de Ciencias Europea y también de la Academia Portuguesa de Ciencias, en la sección de Químicas. Él ha publicado más de 800 trabajos de investigación, tiene numerosos libros, tiene una carrera muy prolífica y tiene más de 20.000 citaciones y además de eso, también compagina esta labor como director del Centro Clínico Egas Moniz de la Universidad de Aveiro, que es mixto, ¿no? Y, en fin, para nosotros es un honor que haya venido y que nos hable de su investigación, que se basa fundamentalmente en síntesis de heterociclos, tanto oxigenados como nitrogenados, y el estudio de su actividad biológica, y también de productos naturales, de su identificación y también de su actividad biológica. Entonces, hoy, como podemos ver, nos va a hablar de química orgánica sintética relacionada con productos biológicamente activos, es decir, con heterociclos oxigenados y nitrogenados. Muchas gracias, Artur, y el floor es tuyo.
Buenas tardes. Sí, ya está marchando. Muchas gracias por la introducción, muchas gracias por la invitación. Es un placer estar aquí entre amigos, porque muchos de que están aquí son mis amigos desde hace mucho tiempo. Está aquí dos personas que cuando llegué a Madrid en 1990 me han recibido, que es Pepe Elguero y Pilar Goya. En el primer día que había llegado a Madrid. A ese tiempo la colaboración portuguesa era más en dirección a Inglaterra, Yo he sido uno de los primeros que he venido para España y para empezar esta colaboración fuerte que ha se fortificado en los últimos 30 años y que ha engrandecido mucho. Agradeceros también el reconocimiento por ser académico correspondiente. Es un honor para mí ser académico de la Real Academia de Física y Química, y os agradecer todo y también por estar presentes en esta presentación. Yo, para hacer la parte científica, me voy a cambiar en inglés porque me protege más en términos de algunas palabras que es más fácil. ¿Sí? Mas me voy a presentar algo que, hablando de la química sintética y de las actividades biológicas que hemos hecho en los últimos años. El primer slide va a ser un poco de atrás, de más antiguos, para mostrar un poco el inicio, mas me voy a intentar no hablar una química orgánica dura porque sería muy aburrida para estar aquí hablando de química orgánica dura. Voy a intentar hacer algo que presente lo que hacemos y por qué hacemos, y voy a mostrar un poco de lo que hacemos en los últimos años. O sea, cuando empecé la carrera, en 1987, cuando Cavaleiro me ha invitado para me quedar en la universidad, porque me he formado como profesor de la escuela secundaria, el tema primero que he trabajado ha sido en cromonas y flavanonas. O sea, había unos productos naturales que habían sido extraídos de unas plantas y ha empezado ahí. So the cromones I have seen the beginning of my academic career, and there is this type of compounds is natural occurring, and I present here some of not only the activity that it's been evaluated, been determined, but also the drugs that are in the market with some of these compounds, and we present here some 2 or 3, and also the package that are in the market. And another type of compound that we have used, we have studied for a long time, is the xanthones. I show here the structure, the simple structure, and also the activity that it's known for this type of compounds. And there is a great number of them. And but there is other applications like food supplements, like we present here. This type of food supplements that is coming from this fruit, this mangosteen, that it's based or have a great number of xanthones in their composition, and they are used in the market as food supplements, as you can see here. And there is other more, not only has a liquid, but also has a solid, a powder, about xanthones of this type that are extracted from this type of fruits, the mangosteen. But well, the beginning, it was this type of structure, is the sterile chromone. So we have a sterile group in the chromone, in the nucleus. So it's this sterile in that part. And it was the first article that I published in 1991. And at that time, when we start, it were known only one compound. It was extracted from South America, and now 9 compounds are extracted and are recognized and are identified from different plants. And we started to look at the establishment of the synthetic methods for this type of compounds. And the first article that we published, it was this one in 1991, chemistry letters from Japan, and it was my baby at that time because it was the first that we published. Of course, then we looked at the biological activity of these compounds. I show here some of the publication that we had, and we had discussed with a company the application of one of 2 or 3 compounds of this type, and we are almost in the application. But then, due to the problems with synthetic aspects, we quit that compounds. But it's why we have this great number of publications in terms of the biological application of this type of compounds. But recently we were not only interested in the compounds themselves, so in the chromones or xanthones, but also in their glycosides. So we want to put some hydrophilicity in the compounds. So trying to put the sugars linked to the compounds. Sorry. And of course that we can have two types of sugars. When they are linked through an oxygen is the O-glycoside, or when we have this type of compound that is a carbon-carbon bond to these compounds. Why we are interested in this? Because of course the O-glycosides can be cutted due to the enzymatic or chemical hydrolysis. And the C-glycoside shows that we have a carbon-carbon bond, is resistant to the enzymatic hydrolysis, so we can combine in the compound the phenolic part and the hydrophilicity coming from the sugar. So this is some of the compounds also. Some of them, like this one that we show here, is already treated or used in the treatment of neurological disorders. So based on that, we try to go in that direction, try to prepare some of these compounds, because we can combine in one compound, so the activities of the phenolic part, the hydrophilic character of the sugar, and we added some hydrolytic stability to this glycosylic bond. So our objective was this one. So try to prepare the two moieties that we can, through a NEC reaction, prepare the compound that we wanted. So we try to prepare allophenolic compounds and sugar alkenes, and then through this prepare the target compounds. Of course that we can, if this is the compound that I will show, is the xanthones with a glycosyl group in different position of that aromatic ring. And of course that we can do it in the final compound. When we have already the structure of final compound, try to do a C-glycosylation at that time, or starting by a compound that you use to prepare that compound. And this is known in the literature, and we tried also to see which is the better way to prepare it. Of course that the first attempt is to prepare the compounds that we needed. So is the sugar with an alkene and the sugar with an alkene. We prepare one with 5-member ring, another with 6-member ring, through the chemical transformation of the sugar moieties that it's well known. So of course that in this case with these two structures, you can see that the last one from the 6-member ring is more convenient, because there are less steps, and at the end we can have more hydroxylic groups. So then, if we have prepared already the sugar, we need to go to the phenolic part. So we try to prepare the xanthone. We started by this fluor benzaldehyde with this phenolic compound. We have the first reaction. It's a typical reaction substitution, aromatic nucleophilic substitution, that the cyclization, and we obtained the 3 compounds that we expected. Of course that when we have, if you look to this compound, when we have the iodine in a specific position, there is 2 possibilities to cyclize, and we obtain the 2 compounds, but it is a typical reaction. So we prepared, in this case, we prepared the xanthones, and we have also the benzaldehydes. And it will be in this case that we will try to do the glycosylation of both. And the next step, it was to do the glycosylation of the benzaldehyde with the 5-member ring and with 6-member ring, and obtain the C-glycosyl without any problems, and with the sugar in all the position of the aromatic ring from the right hand side. So this is a NEC reaction. It's under phosphine-free conditions. We obtain the expected compounds without any problems. There is some isomerization, but I will show one slide to see that it's almost 100% of the one stereochemistry. Then we try to do the same, but with the prepared xanthone. So we have the coupling reaction, the EC reaction, and we obtain the expected compound with 5-member ring and with 6-member ring with the pyranose and furanose from the sugars. And then we try to prepare. So as I established at the beginning, we can do it in this way. We have already prepared the xantone and we do the cyclic glycosylation, or as I showed in the former slide, to have to prepare the C-glycosyl benzaldehyde. So we try to oxidize, to cyclize this compound to obtain the expected xantone, but the reaction didn't occur. So we know at least nowadays that perhaps it's an explanation. The radical that is formed in the acyl group, perhaps it's stable and is more stable than the reaction that we expected. So the compound is not. It's not cyclized, but there is only the oxidation of the benzaldehyde to the carboxylic acid. So it is only to see that the cis isomer, it's only this part, this small part that we have here. So we obtain the compound as we wish in the good way. So then we try to extend the scope of the reaction, and because we prepared xanthones, but if we look now, we prepared chromones. It's in the same way. We prepare starting by the chromone with a bromine in that position and a bromine in that position, and we obtain the expected compound. We can look also to the steryl group, to the steryl chromones. We prepare also the derivative that we want, and we looked at the other molecules that we have in the lab, because we use this as a dye, and we prepared also the sugar derivative. So we can, with this method, we can prepare the C-glycosyl groups in different compounds, and that we can say that using this EC reaction as a key step, we can prepare these phenolic sugars or dyads of phenolics and sugars bonded by a double bond without any problems, extended to the compounds that we want and we wish to do. I will present three parts. It was first part that some work done with the simple compounds from the xanthones or chromones. Now I will look at two different diseases. One of those is the Alzheimer's disease, that it's known very well, and according to the World Health Organization, about 15 million people around the world have dementia, and 60 or 70% is Alzheimer's disease. So there is the different stages or different steps of the Alzheimer's disease. So the first two can be considered non-dementia, and the other ones with dementia. So, and we look to this and try to develop some drugs. So if we look, and perhaps here there is people that knows this better than me, but until now the drugs that are in the market is these ones that are trying to target this cholinesterase. It's a single target drugs. And more recently we have that ones that is based in antibodies, and some of them is to be used in the early stage of the dementia, the Alzheimer's disease, and can have some important results. And why these last ones, or the first ones, the clinical drugs that are available provide only limited symptomatic treatment and do not alter the course of the disease. So it's with this in mind that we start our project. And we prepared some of these compounds, and as you see, it's based on the compounds that will have some experience. So this is the steryl chromones, and this is an extended xanthones, but it's an extended compound. It is flat, and we can prepare this, or we can compare this, that it has some flexibility between the chromone and this part of the molecule, and that one that it's a rigid molecule. And the target, it was to have a multi-target approach, or a multi-target let's say target with or a compound with multi-target approach. Yeah. And we are looking to the cholinesterase, to acetyl and butyryl cholinesterase, and also to the protein aggregation to the A-beta and to tau proteins. And so this is the easy. We established a new method for the steryl compound, steryl groups with this extended that arm that we use in that position that will be used then to prepare the final compound. As you can see, is a xanthone with an extended group there. And we prepare a library of compounds, of course, that I show here only the final compound, but of course we have also the intermediates to have these two libraries of compounds to evaluate the activity. We start by the cholinesterase, the activity to inhibit these two cholinesterase. And the result that we obtained, first we obtained 4 compounds that have presenting only single target activity. As you can see, is for butyl cholinesterase, and this one is for the steryl cholinesterase. In the low micromolar range, as you can see, we have between 4.3 and 9.4 micromolar in terms of the activity that we have. But importantly, we have 7 compounds that present dual target activity for both cholinesterase, they inhibit, and the results it's also in terms of the inhibition, it's in low micromolar range also for this type of compounds. But then we looked not only for the cholinesterase, but we looked also for the other enzymes. But before we try to understand how it fits in the active sites of the cholinesterase. Of course, we can show this in detail, but here we present the 2 of the compounds that present important activity, one from the steryl group, another from the xanthone, and we have what we have seen is that most of the compounds bound to the active sites from the cholinesterase through hydrogen bonds and p stacking interactions. Of course, that we can show in the screen. I don't know if you see very well, but the green is one of the interactions, and the gray is the other interaction. But we show here two compounds showing this interaction in the active sites of this cholinesterase. But then we look to the inhibition of the proteins, the tau and the A-beta. It was the anti-aggregation activity. It was in a single model, as it is right here. It's of course it's not done by us. It is done by my student, but in a group from the Faculty of Pharmacy, and it's with intact Escherichia coli cells that overexpress the A-beta and the tau proteins. And what we have found is that 3 compounds, and as you can see, one is from the steryl group, another is from the xanthones. We have 3 compounds that present single target activity. The results in terms of inhibition, it's good, present good results. I cannot say it's not extremely good now, but it's good in terms of inhibition. But we found that 5 compounds present dual target activity, so they inhibit both the aggregation of both proteins, the self-aggregation, of course, that we see, and these compounds inhibit the self-aggregation of these proteins. So this, of course, that after these results in terms of inhibition, the cholinesterase and also the A-beta and tau, it's important to look at the cell viability evaluation and also to do the BBB permeability. And what we have found is that 4 compounds that is shown in green, this one, the first one, and these 3 below, they cross the brain barrier. So it's a good results in terms of the BBB permeability. And also, if we look to the cell viability, what we can say is that this compound, this 283 and the 285, these are present low toxicity in almost all the concentration that we have used. Of course, that some of the others present low toxicity at the IC50 values, but these two important. It's that two important. The first ones, they present low toxicity at almost all concentrations. So at the end, what we found is that we have 5 compounds that it's emerged from this study that it can be leads for development of multifunctional compounds for Alzheimer's disease. Of course, that there is a mistake there, but it's multifunctional. But here we can see that we have 5 compounds that can be developed for the future. Of course, that we have established the structure-activity relationship, and we can see how it is with the groups that increase the activity in terms of cholinesterase, or those that increase the activity of the inhibition of the tau and the beta protein. So it's a result that we have, but the most important is that 5 compounds that we obtained, and it can be leads for this type of activity. Following our interest in the Alzheimer's disease, we have other small molecule inhibitors for the protein aggregation. And it is very well known that since 2015 and 18, with these publications in Nature in JAMA Medchem, that the lanosterol and the derivative of lanosterol inhibit the crystalline protein aggregation from a mutant of the crystalline aggregation of the crystalline protein. So this is important. It was important for us to start our study. And the lanosterol in 2019 appeared also to inhibit the aggregation of the amyloid beta. So this is the 3 results that it started. It's why we start our study in these compounds. Of course, that if we look very, very deeply in the publications, we can see the inhibition of the lanosterol, the inhibition in the A-beta aggregation, and also the cholesterol. And we can see here also the comparison between the lanosterol in different concentrations, in the cholesterol also. But there is also one compound here that it's a phenolic compound. It's not a xanthone or a colomone, but it's a phenolic compound. And if we look to the inhibition, is the compound that presents the better results is the green one. So it was the beginning to look at different compounds for this type of activities. Of course, that if we look also to the self-aggregation, we can see the control, the cholesterol with 500 micromolar, the lanosterol with 200 micromolar, and the lanosterol with 500 micromolar, and the inhibition, as it is here in the results, we can see here in the picture very, very, very well. So for us, we looked at try to combine in the molecule the cholesterol and the quinoline moiety. Why we looked at this? The cholesterol having hydrophobic interaction with A-beta. It have BBB permeability, and of course it's a cheap starting material if we compare with lanosterol. And in terms of the quinoline, it also interacts with A-beta, is ionizable scaffold, and it's a good template for near-infrared frequency for theranostic. So we try to combine these 2 molecules. And we will try to prepare this we call linear compound, and this one that we call angular compound. So in terms of synthesis, we start by the cholesterol. So we perform an oxidation to have this alpha-beta unsaturated system, and then with the amino acetophenones, with this Friedlander annulation, we prepare this angular derivative combining the cholesterol and the quinoline moiety. Then, if we do a different oxidation, not only to have an alpha-beta unsaturated system, but to have this diketo unsaturated system, and then the next step, it will be the combination with the amino acetophenones to prepare the linear derivative as we wanted. Of course, that we have prepared 2 small libraries for compounds for each one of the libraries, and then we look to the activity, to the inhibition of the capacity to form fibrils with the habitat. And if we look to the compounds from the series A, one of these compounds present a very important activity. The other ones has less inhibition activity, but the 4D present an important activity. And we can look at the structure, and this one, it's the compound that presents is the 4D that presents the activity. It presents an inhibition of around 75% at 20 micromolar in terms of inhibition. If we look to the series 6, that it's the linear one, you can see that the derivative is 6A, the most important compound in terms of inhibition of activity. And in this case, we reach around 87% at the same concentration. So it's important results in terms of the inhibition to the formation of the fibrils. Then we try to use the same compounds for disaggregation, the capacity of to have the plex. So because there is a pre-aggregation synthetic peptide, and then we try to deaggregate, to do the disaggregation with our compounds. And as you can see in the here, the compounds were able to decrease the number of foci to basal levels, save this one, the 4C and 4B, that increase the number of foci that we compared with the self-aggregation of the beta that we have. And another important aspect that we must look is not only to the number of foci, but also to the area of these foci that we have. And in the right-hand side, we can see that some of our compounds, not all of them, but some of our compounds were able also to significantly reduce the area of the foci that we have. Of course, that we can see this also by the picture from the microscopy, the confocal microscopy applied for these two compounds, but it's only to show you a little bit that we have seen this also. We have also to try to see this disaggregation capacity when do we induce the aggregation by the needle TNIP. It's a way to aggregate and then try to use our compounds. And once again, we can see that the number of protein aggregates decrease with our compounds, and the areas too. There is one compound that is an exception, but all the other cases also, the average areas of the protein aggregates diminish with the presence, with the activity, with the presence of our compounds. So we have seen also by the microscopy, the confocal microscopy, the same for two compounds that it was presented in the former slide. So what we can have as a, let's say, as a conclusion, this is when we start this, it was in the market, what we have in the market, let's say in the literature to present the cholesterol, and in our case we reach up to 87% of the inhibition with 20 micromolar, and some of generally our compounds reduce the aggregates to basal levels and reduce the area of this foci also. So there is some discrepancies in terms of the number and the area of the aggregates that suggest some selectivity of our compounds, and we want to go a little bit further to prepare other derivatives and try to have a structure activity relationship in terms of the activity, but the results are promising for us. Yeah. So another, it's following the same field, we try to replace now quinolines by a more soluble moieties. So we start by the cholesterol. It's one of the compounds that we prefer in the former slide, and then we have done an aldolic reaction. And although there is two possibilities, the position 2 and the position 4, and it was exclusively done in that position. And then the last step, it is the reaction with the malononitrile, and we obtain this pyran fused cholesterol. And if we use a bigger excess of this malononitrile, we obtain this type of compounds, not a furan ring, but this benzene ring. So we obtain this amino isophthalonotriles, and it's also an explanation how we can prepare these compounds. But the important one is to have this family of derivatives, and we will use them to try to evaluate the activity, to inhibit the enzymes, but also try to disaggregate the aggregates that we have in the Alzheimer's disease of the proteins. The last part, it's about another disease, is the anti-leishmanial or anti-leishmaniosis. And why I present this, because in the first study about the Alzheimer's disease, it was the compounds that we prepared. It's based in the intuition, well, let's say, it's empiric. We have combined the cholesterol and quinolin. Here we have a different approach. So we have the leishmaniasis, is a neglect disease from the 21st century, typical from tropics, subtropics, and some parts of the Mediterranean basin. But the study that we have done, it's based on virtual screening. We start by a virtual screening and molecular docking. Then we prepare the compounds. Then we evaluate the biological activity. And then we looked once again to the molecular docking to see if the compounds are how we fit in the active site. Why we looked at this disease? Because as it's written here, because the actual medicines present limitations, both in terms of toxicity and affordability, and it's become mandatory to develop some of the anti-leishmanial agents. This is the establishment of the why we looked at this, and the important is that the two last lines, 2.5 million new cases each year, and the mortality rate is about 100 people person per year. So it's a big and a complication. So this the leishmaniasis is based in the leishmaniania parasite. It's transmitted through a bit of the infected females, and there is a lot of species of these females. And the disease is characterized by a wide range of clinical manifestations presenting distinct degrees of complexity and severity. We can have type visceral, cutaneous, or mucocutaneous, and this is the most common clinical forms of the leishmaniasis. So there is no vaccination. As I referred before, this is the drugs in the market nowadays, and as I referred already, present limitations, like it is an elevated cost for patients, is loss of efficiency of the drugs, there is the development of resistance mechanisms, and there is the appearance of several adverse effects. So it's, as I referred, it's mandatory to look at this disease. For us, we started by a virtual screening. Then we tried to develop the synthetic methods based on the principles of the green chemistry, and I will refer at the end why. So there is Leishmania pieris. It's a highly complex parasite. It have here a considerable variety of metabolic pathways when we can apply. And we choose on different enzymes we can be the target for our studies, and we looked at that enzyme, that the S-adenosyl methionine decarboxylate. Why? Because this enzyme is responsible for, has a crucial role in different aspects that are related with maintaining the potential of the membrane of the cell and also controlling the pH and the volume of the intracellular pH and volume of the cell. So it's why we looked at that specific enzyme. So we started by 100,000 compounds to do virtual screening. Then from there we have 250 promising molecules. We need to analyze them because some of them is impossible to synthesize when the computer has crazy things. And then from the 250 we choose based on the quinolin and the ecridine, we choose 6 the most promised ecridine based compounds. And this is the structure that we start to look for our synthetic methods. Of course, then we look to prepare the compounds. We start by single molecules, and the first studies is to prepare the building blocks, to prepare that molecules. It's a little bit crazy, some of the molecules. So we prepare the compounds. In some cases, we use always the microwave to have the transformation in shorter reaction times, because if it is not with microwaves, the reaction took more than 10 hours, so it's a way to have shortened reaction times. We obtain these compounds without any problem, in good yield, but we must be careful about water, because if we have some water in the reaction, we obtain not the chlorine, that position, but hydroxy derivatives that are not what we not expected. The next step it was to add this diamino derivative that we want to add at that position. We need to control very well the reaction, because if not we have other compounds, because as you can see we have a diamino group, so we can have dimers and we want to have only monomers, so it's necessary to control very well the reaction. And we have done this with these 3 rings, it's the ecridine derivatives, and with these quinolin derivatives to prepare also the derivatives that we want for the next steps. The last step is that one is to prepare the final compound, the 1,3,5-triazine derivative. So we start by this compound, the cyanuric acid, and we replace one of the chlorines at a time. So first with the building block that we prepared, with one specific aniline, and then with several nucleophiles to put at that position different nucleophiles. So we prepare this small library of compounds. Then we look to other derivatives, in this case with quinolin, and we prepare also another library of compounds. And in some cases we try to, if here we use with this aniline, then we use another aniline. So we try to combine different parts of the molecule to have at the end libraries that can give us some insight about the activity of these compounds. Then we evaluate the biological activity. The Leishmania parasite is an organism that exists in 2 distinct forms. One is the extracellular, it is called promastigotes, and the other one is inside of the cell, is amastigotes, but we evaluate this as a macrophage because it's cells that are prone to be infected by the promastigotes. And this activity was done in Porto, in the Institute of Research for Innovation in Health, and it was done in collaboration, my PhD student, with the colleagues from there that have the experience on that activity. So as we can look, here, this is one of the building blocks, and that we have the activity of the building blocks. We have also the activity of the drug that it was used as a standard for our compound. As you see, our compound presents an interesting activity compared with that one. And from our library, only that derivative, it's the important. It presents important activity in terms of the promastigote activity, but also in terms of the cytotoxicity and therapeutic index, also so related with amastigote. So from this small library of compounds, only one, but if this one, it's present good results compared even with this, the drug that we use as a standard. The other derivatives, it's less important, the activity, because here it's almost some of them is toxic, but there is no important in terms of therapeutic application. In terms of this, the quinoline derivatives, we have 2 compounds that present important activity for the future. So in this part, we can look with this structural activity, we can see some of the important aspects that we can look to the future to improve a little bit the activity. And if we look to this one, we can see, as it is written here, is small groups like methylamine and methoxy groups leads to increased activities. Is this the important conclusion? And in this derivative like this one is the diamino aniline group that it's considerable increase the activities that we have looked at this part of the activity. Then, of course, we do the redocking of the compound that we prepare to see if the virtual screen activity it was a good insight for this. And of course, we found that in the allosteric site of the enzyme there is important interactions, most devoted to hydrogen bonds and also PP interaction, and it's only the pictures want to see that it fits quite well in terms of the allosteric center of the enzyme. Then one of the compounds, it's based to have, instead of being all the aromatic, all the 3 rings to be aromatic, we have one of those, it's saturated, it's the tetrahedral derivatives, because one of the compounds in the virtual screen it shows also that it will be an important compound. So we prepared the compound that we wanted, once again the acridine derivative, in this case with the diamino, but the alkylic diamine group, and we prepared the 2 families that we want, the libraries. But as impurities, let's say, or byproducts, we obtain these dimers, and we isolate them, and then we improve and we develop a method to create to synthesize these compounds, and then to evaluate the activity of them at the end. So we have done more to be in according to the virtual screening. It was necessary also to acetylate the amino group, the final amino group. It was easily to prepare, and there was also one of the compounds that involves dimers of this, but at the middle having a basoquinone derivative. And well, we have prepared this, but in this case we can have 2 additions at that position without the chlorine, and in that position with the chlorine. So in each one of the reaction we obtain 2 compounds, one without chlorine and other with chlorine. And we prepared also these compounds, and we look then to different combinations to have the compound that we expected. But one of those, it was, we wanted to prepare to have a combination of the, let's say, acridine derivatives with chromone derivatives, because it was also one of the compounds involves both of the structures. But unfortunately, the reaction didn't occur, and we obtained a byproduct that we obtained. But after several attempts to combine these 2 compounds, it was not, we were not able to prepare the compounds. Unfortunately, in terms of application, the compounds present high toxicity, but in terms of therapeutic application, there was no good results for none of these compounds. And of course, that now we are trying to look at the structure to see if we can adjust the substituents to have some therapeutic application, and not only the toxicity that we have in this type of compounds. Of course, that we, from our libraries, we can also have some structure activity relationship, and we can see in which are the groups that are important to increase or to decrease the activity, but for the moment it's only toxicity and not the therapeutic applications. We have also do some redocking about the structure that we obtain in this case. There is also a good fit and some interactions, hydrogen bonds and PP interactions, so that we can show also that it's the virtual screening, it was good in that way. So this is a different rationale that we are doing virtual screening, synthesis and biological application, biological activity, and at the end to see making the redocking to see if all the things fit. We obtained some compounds, as I will show here. So we start by 100,000 compounds. From there we obtain 250 compounds that can be prepared. From there we choose only 6 compounds, 6 structures. From the 6 structures we prepare 72 because our derivatives are homologous. From these 72 compounds, 20 compounds present important promastigote activity, anti-promastigote activity, because it presents an IC50 of less than 5 micromolar, and the microphage activity from that 20 compounds, 5 compounds present important therapeutic index that can be further studied, and this is the compound that we have, and also the final structure, and this is a way like now in our group we look at the synthesis. We are doing first the virtual screening, synthesis, biological evaluation, and to see in this way is not only empiric but it's based on a target that normally is, as you know, is a protein. Finally, I would like to thank all the people that was involved in this work, mainly the students that were with me and some of the postdocs, and we see here now the position that they have. Also some collaboration from our universities, from the health department, and here some collaboration that it's from Portugal and Spain. Some of them it's a long way. By starting by Professor Alario when we started in 1990, and I would like also to thank the people that give us some money. And as I referred in some part of my talk, we were looking now to the principles of the green chemistry, and my research center is called, in my affiliation is this one. This is in Portuguese means associate laboratory in green chemistry. It's Química Verde. It's a research center that involves persons from the University of Porto, from several faculties, from the University of Aveiro, our group, from the new University of Lisbon, and in total we are 1,100 persons. And we now, when we do a meeting, it's only a virtual meeting because we don't have a facility to put all together. All together it means professors, researchers, PhD students, and so. And normally we do this. The meeting is only for invitations for 200 or 300 people or colleagues, and the others attend the meeting by virtual meetings. So I would like to thank you all for attending this session where I tried to present some of our activity and I tried to avoid the organic chems details. Thank you very much.
Gracias. Muchas gracias, Artur, por esta colección, estas librerías de compuestos, todo el trabajo sintético y de actividad biológica que habéis hecho. Y realmente hace falta tener un grupo enorme para poder hacer tantísimo trabajo, ¿no? O ser muy eficiente, evidentemente, pero ha sido un trabajo también de muchos años, supongo, ¿no?
Sí.
Pero ha sido brillante todo lo que habéis conseguido y la verdad es que es muy complicado, ¿no? Bueno, es momento para hacer preguntas por parte de la audiencia.
Thank you, Artur, for this beautiful heterocyclic chemistry you show with us. Beautiful examples. Some of them are relatively close to me because during my thesis time I was very much engaged with 4H, 2-amino 4H pyrene rings. And you use this heterocyclic ring just to carry out some derivatives with steroids, I guess. And however, under certain conditions, you can go either to the pyrene ring or to the carbocyclic ring. However, I don't know if you just to bring to your attention that the pyrene ring is a highly reactive system. So instead of using, for instance, malonitrile, if you use cyanide, you are able to get instead of the pyrene ring to the furan ring, or you can transform the 4H pyrene ring into a nitrogen ring as well. So it could be easily expanded, the number of systems they are related with this highly reactive ring. So this is an interesting suggestion because that chemistry is very well known, so I'm pretty sure it would work also in your case. This is just a comment, but just going to your systems, I could see that, I guess that the biological studies you carried out were in vitro yes. All the times, and however you are moving usually at the micromolar concentrations for the EC50 value. And it is today, I don't know, because you show it that many, but is competitive, these values, or are at the nanomolar concentration or even smaller?
So we in some cases we don't present the best structures because you need to keep them from the, if you want to patent. It's why some of the structures are not here. So because we have some of them in the nanomolar concentration that is the IC50, it's in the nanomolar. It's not so much structures we have in the disaggregation. We have the three structures that we have in this range of the concentration. While in the leishmaniosis, it's the activities is competitive with the drugs that we have in the market because it's very bad the drugs that are in the market.
Okay.
In terms of side effects and in terms of resistance and so on.
Yeah, I have a curiosity. May I go to another question?
But a comment in the structures. Yes, as we have done with malonitrile, we have already started with other methylene active compounds, and we do the addition and then the cyclization with different compounds. Not only with malonitrile. I present here only malonitrile, but with different methylene active compounds we can put some interesting structure there. Yeah.
Okay. Even ring contraction to carbocyclic systems by photochemical procedures can be also carried out. Did you go one step further to other in vivo experiments in any of your systems?
Yeah, it's I have a collaboration with South Korea. It's a colleague that it was with me in an European project, and 10 years ago, and we still maintain the collaboration. And now he's looking at the in vivo activity of some of these compounds. We have also with him some of the chromones, and there is also some compounds that combine chromones and pyrazoles. That's not the objective of this. And we gave him also The last, it was 3 grams of compound, because they need, no, it's not only the milligrams, because when we go for the in vivo, it needs 3 grams. And I hope that we can reach some interesting results, but for the moment it's in South Korea, yes.
Okay, so thank you so much and congratulations for your entrance to the Academy.
Yes, thank you very much. Very interesting. I have a question related to the Alzheimer's disease and the activity on the anti-aggregation. So if I understood correctly, and maybe I'm totally wrong, but if I understood correctly, when these proteins aggregate, it is because they somehow get scrambled, so they lose somehow the conformation that they have in the natural state. Is this something that you also take into account when studying the anti-aggregation effect? And do your compounds help us understand which are the parts of the protein which actually promote the aggregation?
No. When we start, it was based on that compound lanosterol. Lanosterol, when we look at lanosterol, we were starting a project on the Alzheimer and neuropathological disease in general, and we look at that structure and say, why, if it present good results, lanosterol, why not to try to do this? And of course, that there is some discussion about when do you disaggregate. That he says that the small aggregates is more toxic than big ones or so, but it's a way to look and try to do. But no, there is no information about the part of the enzyme. No.
Yeah, thank you.
¿Alguna pregunta más? Ivonne primero.
Gracias. Te voy a hacer la pregunta en castellano.
Sí, puede hacer sin problema, sí.
En el proceso que seguís de investigación, en el caso de elimination, hacéis un virtual screening que asumo que es similar al docking, al redocking, que es al final. Entonces, hacéis el virtual screening, seleccionáis una serie de moléculas, las sintetizáis, las probáis y volvéis a demostrar que esas moléculas encajan bien. Si por lo que sea hay un problema sistemático en la modelización que hacéis, estáis perdiendo un conjunto de moléculas que evidentemente las que habéis seleccionado sí que van a encajar, pero hay otro conjunto que puede ser. Entonces, no sé, quizás es una cuestión a pensar, ¿no? Cómo es el primer paso y es el paso quizás más importante que te va a guiar en todo el proceso, ¿no? Entonces, habría que buscar algún tipo de test para comprobar que ese primer paso está bien hecho.
El virtual screening.
Sí. ¿Entiendes, no? Lo que te
quiero decir. Sí, porque si no, al final, cuando haces el redocking, en principio debe funcionar, porque al principio lo has testado y ya ha empezado por ahí. Sí, es perfecto, sí. Sí, lo primero paso, sí. Mas ahí es mi compañero de química teórica de Porto que mas podemos estar a perder un conjunto de moléculas que si haces un pequeño error ahí...
Sí, que puede ser que el modelo no reconozca bien la interacción de un grupo con parte del receptor o lo que sea, y entonces pues esas moléculas quedan automáticamente fuera.
Sí, y después, como sabes mejor que yo, es basado en la literatura que tienes el X-ray de la proteína para hacer los cálculos. Puede tener ahí algunos problemas al principio también, sí. Mas sí, ahí todo el otro era empírico, o sea, era la química, la tu idea, que aquí te baseas un poco en el ordenador, que tienes la estructura de la proteína y ver si encaja ahí, mas sí, si tiene un problema ahí, todo el resto tiene sus problemas por encima, sí.
Muchas gracias, Artur.
Mi cuestión es muy general. Vosotros observáis que cuando hay cloro en la molécula o no hay cloro en la molécula se comporta de manera diferente. No muy diferente, pero diferente. Y mi pregunta es, ¿y si hubiera un flúor?
Mas en muchas de las
En
moléculas...
vez del cloro, si hubiera un flúor.
Mas en muchas de las moléculas ahora vemos específicamente colocado un flúor.
Ajá.
Y sí, lo flúor te da una actividad interesante. Mas yo no tiene nada que ver con esto. Mas, por ejemplo, cuando piensas en actividad antioxidante, si tienes o no tienes un cloro es muy importante. Habíamos llegado ya a eso, o sea que si tienes un buen antioxidante, que por tener los hidroxilos, por tener lo que sea, y se introduce un cloro o dos cloros, la actividad es muy más importante. Mas aquí, en muchas de las moléculas, tiene, por ejemplo, la parte de la leishmaniosis, nos habíamos colocado mismo la fluoranilina. Y en varias de las moléculas habíamos colocado también flúor, sí.
Gracias.
Ahí lo del flúor tuvimos aquí a Macmillan hace poco, que ha sido el que ha desarrollado los grupos CF3 prácticamente dentro de la química médica. Tengo que decir que el grupo CF3 no existe en la naturaleza.
Sí, no voy con el CF3,
ya, un solo flúor, pero eso por ponerlo como precedente. Sí. O sea, el CF3, que es bioisóstero con el grupo metilo, pero que no existe en la naturaleza, sí, y sin embargo tiene un efecto en algunos fármacos muy interesante, el CF3. El flúor es verdad que sí existe en antibióticos y en muchos más, pero en algún caso de los que has puesto en el cloro, por ejemplo, el 1,5-dicloro para benzoquinona que has puesto, ese si fuese flúor en vez de cloro no funcionaría, porque estás llevando a cabo la reacción química sobre el cloro, y con el flúor no funcionaría porque el enlace carbono-flúor es muy diferente.
Sí, sí.
Bueno, yo tengo una pregunta.
Una curiosidad, ¿es un problema de salud pública la leishmaniosis en España y Portugal?
No, no.
Cuanto más al sur, más prevalente, ¿no?
Sí.
En África, en concreto. Sí, sí. Tiene que ver con
En Portugal y España son animales. Y animales sí existe.
¿En él?
Perros.
Ah, ya.
Los perros, sí. Bueno, yo tenía una pregunta de alumna, pero creo que todavía no hemos acabado, así que yo me la reservo para el final.
Gracias a tú por la charla. Así hemos visto todo lo que has hecho durante todos esos años, o una parte por lo menos, ¿no? En la parte de las series eso de anti-leishmaniosis, tienes compuestos que son quinonas. ¿Habéis pensado hacer antitumorales, probar algo o lo que sea? Y si no, pues en el instituto tenemos ensayos.
Sí, hay una persona que ya ha visto mis estructuras y ha pedido si lo quiere testar, sí, porque sí, serán tal vez buenos compuestos, aquellos 4 al final derivados de ellos, sí.
¿Alguna otra pregunta? Bueno, pues yo voy a acabar haciéndote una pregunta casi de alumno o de alumna. Cuando introducís el grupo estirilo en los cromenos o cromanos, hay posibilidad de que, bueno, aparentemente sale el trans, pero tenéis un aspecto conformacional, que puede ser S-cis o S-trans. Tú nos has pintado en general la conformación S-trans. Entonces, supongo que habéis determinado por resonancia que la molécula se encuentra justamente en esa conformación.
Por ejemplo, Pepe se debe acordar de eso, y por rayos X también.
También.
Y es la forma más usual. O sea, se puede preparar también el cis, más con luz. O sea, en la fotoquímica se puede hacer el compuesto en solución, se le metes luz, puede ser luz del sol, se va a isomerizar y te puedes... llegas a una mezcla de 1:4.
Ajá.
Un cis y 4 trans, más normal, se hace la síntesis y es siempre trans, la doble lesión y ese trans para arriba, sí.
Ajá, muy bien. Voy a ver si hay alguna pregunta de la audiencia que tenemos a través de YouTube. A ver si ha llegado alguna pregunta. Bueno, yo creo que son sobre todo felicitaciones y saludos. No hay ninguna. Entonces, como no hay ninguna pregunta, vamos a dar por terminada esta parte de la sesión. Felicitar de nuevo a Artur y agradecerle su charla.
Muchas gracias.
Pues ahora viene la segunda parte, que es la más interesante, claro, que es ser académico correspondiente y darle su diploma, pues lo da así. Entonces, aunque no soy el presidente, sino el presidente suplente, pero es un honor para mí concederte darte el diploma correspondiente. Y ahora tenemos aquí el libro de honor y luego esta foto.
Esta foto
una
que la tienes que
sí.
dedicar a
que
la
va a firmar
academia.
dedicando a la academia una cosa, si quieres te sientes.
Sí, sí.
¿Quieres sentarte?
De acuerdo, sí.
Vamos a subir a ver las instalaciones.
Sí, sí.